Hello, my name is Matthew Magee, and I will be presenting my research on alternative N-termini splicing of the B52 protein in Apis mellifera flight mussels during the nurse to forager transition. Female western honeybees, or Apis mellifera, exhibit a type of phenotypic plasticity called polyethism during their transition from nurses to foragers about 3 weeks into their lives. During this transition, nurses develop thicker flight muscles, increased aerobic metabolism, and loss of mass, among other metabolic, neural, and behavioral changes. These changes are partially regulated by the splicing mechanism, which includes 6 small nuclear ribonucleoproteins, or SNRPs, seen here in light green, and the formation of a lariat, seen here in yellow, to create a mature mRNA. Alternative splicing also helps regulate this transition through intron retention, alternate 3' and 5' splice sites, exon skipping, and others, allowing for many proteins to result from a single pre-mRNA. B52 is an SR protein found in flight mussels that helps guide the U1 and U2 SNRP complexes to the branch site to initiate splicing. Seen here is a proposed model of B52 that is also seen in Drosophila melanogaster. SR proteins have an SR domain heavy in arginine and serine amino acids that helps recruit splicing machinery via protein-protein interactions. B52 also has 2 RNA recognition motif, or RRM, domains to bind to enhancer sequences and a glycine-rich hinge between these RRM domains to allow for flexion and specific binding of the protein. This research focuses on alternative splicing on the N-terminus of B52, which is at the first RRM domain, and how these isoform levels change as the nurses transition to foragers. The methods for this research are fairly straightforward. Flight mussels were removed from honeybee thoraxes and RNA was extracted from those mussels. Quantitative PCR was used to analyze the data, utilizing cyber-green dye to bind to double-stranded DNA and reflect amplification in real time. RPL8 is a protein found in the 60S ribosomal subunit and was used as a constitutive quantitative control. B5256R primers amplify 2 constitutive exons to measure total B52 mRNA levels, while 2F2R and 34R are primers amplifying different N-termini start sites. The results for the nurses show no significant difference in 5,6R levels, as total mRNA should be the same for all samples. For 2F2R, further analysis is necessary to determine if these differences are statistically significant, but there is definitely more variation compared to 5,6R. There were only 2 usable samples for 3,4R due to time constraints thanks to COVID, so we were unable to make conclusions on that data at this point. 5,6R was then normalized to 2F2R, but again, there were only 2 samples, so conclusions can't currently be made. However, it does make sense that N10 levels were higher in this analysis because N10 levels were also higher than N9 levels when normalized to RPL8 in the other 3 analyses. The Forager dataset isn't as complete as the nurse dataset, again due to COVID, but there are some results that can be taken away from it. In 5,6R, there are a few samples with significantly different total mRNA levels. Although there isn't a direct correlation of increased mRNA levels with age, one possible explanation for these differences is that some bees may have transitioned into foragers earlier and had been flying for a few days before capture, while others transitioned late and had only been flying for a few hours or even minutes before capture. There's no real way to regulate that. For 2F2R, there is a significant difference between F8 and F9, which happen to be from different samples of the same age. If you combine the data from 5,6R and 2F2R, you can see that the percentage of 2F2R isoforms in the total F8 sample is much higher than the 2F2R percentage in the F9 sample. This research is important because it looks at the various isoforms of honeybee B52 mRNA, both beneficial and poisonous. Beneficial B52 isoforms contain 2 RRM domains and 1 SR domain and are able to fully initiate splicing. However, poisonous isoforms are missing at least one of these domains and can only bind to the splicing machinery or the pre-mRNA and are unable to initiate splicing, taking up SNRPs that could be used by full isoforms to actually splice and create mature mRNAs. For future work, more samples of young nurses and foragers will be used in qPCRs to complete the datasets. Once analyses are run on these datasets, the next step is to use the same primers on older nurse and forager samples. Finally, moving to the C-terminus and focusing on the SR domain as the last part of this project. I would like to thank the School of Science and Math, the Biology Department, Dr. Agnes Southgate, and IRCA for allowing me to conduct research while in college. Thank you for your time.